As we know, DNA is primarily what makes up almost all organisms' genetic material. Now, within DNA, it can go through a type of clumping, and we've referred to that as developing some chromosomes. So if we look at the overall structure of DNA, which once again, it can be in a nucleus if you're talking about eukaryote, or it can just be in a what's called a chromosome in a prokaryote or a genome, what will happen is the DNA will start to crumple together. And what it does is it's held together by a series of DNA as well as proteins that we refer to as histones. And that's what gives it kind of that bulky appearance. Now, normally the DNA will just kind of be all over the place, but if we, for example, talk about prophase, or if we talk about binary fission with prokaryotes, the chromosomes and DNA will start to condense. But just realize that the chromosome is just a segment of DNA as well as proteins that work together and give it that X-looking structure. If we talk about a particular gene on a chromosome itself, right, if we have a chromosome here, a gene is a certain portion of the chromosome that might code for hair color or eye color or something like that. So that's kind of the relationship. Once again, chromosomes are made up of DNA as well as proteins. A gene is a certain segment on the chromosome or a certain segment of the DNA. Now, if we start talking a little bit about the overall structure of DNA, it is what we call a nucleic acid. And the building blocks of nucleic acids are nucleotides. Each nucleotide consists of three parts: a sugar, a phosphate group, and a base. Now, we know depending on the nucleic acid, it might be a different sugar, there might be different bases involved, but we'll talk about those a little bit. So this is one of those molecules of life. The other three, of course, being lipids, proteins, and carbohydrates. In DNA, there are four bases: A, G, T, and C, adenine, guanine, thymine, cytosine, and those are the ones that will make up the middle structure of the DNA, as well as having the phosphate groups and the sugars that will make up the backbone. Now, we've talked about this as well, but just a quick review. If we talk about the overall reasons why A has to bind with T and G with C, there are two main reasons. First of all, when you have DNA, you want to maintain a 3-carbon ring in the middle. So if we add these up, obviously we have 1, 2, and 3. If we add these up, 1, 2, and 3. Now, that's one of the reasons. So we wouldn't want to have A with G, because that would create a 4-carbon, and we wouldn't want to have cytosine with thymine because that would create a 2, so go 4, 2, 3. Our DNA would almost have a lumpy appearance. Now, if you have the question, well, why can't cytosine bind with adenine up here? The other reason why you have to have A's with T's, G's with C's is because of the chemistry. Certain things just don't cause a reaction with each other. You don't have a reaction with everything that you mix, and that's the case with these different bases. So with adenine, binding with thymine, it creates that 3-carbon ring, and also it allows them to create a hydrogen bond. That's what holds the DNA together. Same thing over here with guanine and cytosine. We maintain that 3-carbon ring in the middle, and it will also form hydrogen bonds. So that's the overall structure of DNA. All right, another thing that we're going to talk about real quick here is the difference between DNA and RNA. Once again, they overall have the basic structure of all nucleotides. So they have a sugar base and phosphate groups, but there are differences between. So overall, if we just look at the picture here, notice how DNA is on the left, RNA is on the right. We know this because we have a double helix structure. Gets its name because it has 2 strands and a helical structure because it kind of looks like a roller coaster. Where if we look at RNA over here, it is a single strand molecule. So that's one of the differences between DNA and RNA. The other difference is the bases that make them up. So in DNA, we have cytosine, guanine, adenine, and thymine. Sorry if you can't read it. For RNA, we have 3 of the same. So we have cytosine, guanine, and adenine. The key difference, though, is that we have uracil, the U, instead of thiamine in DNA. So that's another difference. And finally, if you look at the overall names of the sugars that make them up, we have DNA. It stands for deoxyribonucleic acid. RNA is ribonucleic acid. So what this literally means is DNA lacks an oxygen in the sugar that makes up the backbone. So three differences: double strand, single strand, we have a T instead of a U, and we have different sugars, deoxyribose sugar and just straight up ribose sugar. The differences between DNA and RNA. All right, one of the things that you guys wanted me to go over was the different processes that happen as far as how the cell duplicates or creates new. So let's first start off with just talking about the cell cycle. We've seen this before. We are going to start off in this corner where we have the official kind of birth of a new cell. Right after a cell divides, it will go through what's called G1, which stands for first growth, where it's going to just become kind of its everyday size. Then it will go through a S phase. This S stands for synthesis, where the DNA is going to be duplicated for the next time that it divides. And then also it will go through a second growth, and this is a time where the cells will actually be a little bit larger than normal because it's prepping to divide. Collectively, if we look at this, G1, G2, and G1S and G2 are collectively called interphase, and they take up about 90% of the cell cycle. So most of the cells that we look at underneath the microscope are just going to be in interphase. Now, once we get closer to the division time, if we break down this cell cycle, here we have the process of mitosis, which includes prophase, metaphase, anaphase, telophase. We'll get to those in a little bit. And this last little sliver right here represents cytokinesis, where the cells will officially split. Now, we've also talked about how within the cell cycle there are going to be various checkpoints. Those are important because what those checkpoints do is they monitor to make sure that the cells are healthy, that they have the appropriate amount of chromosomes, that they're the right size. If there is some type of mutation in the proteins that actually do the checkpoints, sometimes cancer can occur. And we define cancer as an uncontrolled growth of cells. So if they are not monitored at the different checkpoints, so there's a checkpoint after G1, after the S, and then also after the M phase, you can sometimes get a result of cancer. So that's a cell cycle. We'll get into further now the process of mitosis and how the cell will actually divide. We are now going to go through the sequence in which our cells divide. This is on page 146 of your textbook if you want to follow along. But once again, here's kind of where we're going to start off with. So interphase is what we just talked about. Once again, that's 90% of the cell's life cycle. Once the cell is deemed to divide, it will then go through what we call prophase. Now, sometimes your book will get a little bit fancy in calling it early prophase or late prophase, but here's kind of the key concepts to this. When prophase takes place, what you will start to see is the nucleus chromosomes are going to start to condense. So instead of just looking like a whole mixture of stuff, it kind of looks like spaghetti as those chromosomes are starting to condense. The other thing with this is you will also start to see the centrioles go to either sides of the poles to kind of help out with dividing and organizing these chromosomes. So once again, here we start to see the actual X's starting to condense into chromosomes. As we continue on to what some textbooks will call prometaphase, this is kind of the mixture of prophase and metaphase. Here you will start to have the chromosomes migrate to the equator of the cell. The spindle fibers here will be attached to the centromeres of the chromosomes. And then finally, when we officially say we're in metaphase, that's when the chromosomes are going to be at the exact center. Once again, if you use the little word hitch, metaphase middle, that will help you out. Chromosomes are going to be lined up on the middle. Once those chromosomes are on the equator, they will now be fully attached by these spindle fibers, and kind of like a fishing pole and line, the anaphase and centriole portion of this cell division is going to start to reel those chromosomes in. So during mitosis, it is the X's, the chromatids, that are going to be split. Okay, contrary to meiosis, we'll get to that in a little bit. But here you have the half-X's, also known as chromatids, actually doing the splitting. The little helper with this is these chromosomes kind of look like the letter A. A is for anaphase, once again, if that helps you out a little bit. Finally, once the chromosomes have officially separated to either poles, it will go to the process of telophase, telophase 2. So these cells will actually have 2 nuclei within them. This is sometimes what you'll see right before the cell is like the same size, but it's got 2 nuclei, and that's the key to telophase. The chromosomes will also start to decondense, kind of go back to the regular looking nuclei. And then finally, when we get past telophase, we now have cytokinesis, and then interphase will start once again. Once again, cytokinesis just means the official splitting of the cell. Now, this is the overall sequence of the animal cell division, but with plant cells, they will have an additional type of division, and that is they will have cell plate formation. Because they have the cell wall, they'll have an additional cell plate formation step where that will need to, bring all those fibers together, all the cellulose together, and create the cell wall. Okay, so once again, we have interphase, that's 90% of the cell cycle. We have prophase, we have metaphase, we have anaphase, telophase, and then finally cytokinesis. That's the steps of mitosis. We are now going to take a look at an interactive file here that shows the differences between the process of mitosis and meiosis. So on the left, you'll see the process of mitosis. Some keys here are this cell will go through asexual reproduction. It will create an exact copy of itself, or a clone. So we'll start off with 46 chromosomes and finish with 46 chromosomes. Net results are 2 diploid cells. We'll go through one sequence of the processes of, or the stages within mitosis. If we go to meiosis, here we'll get a little bit of variation through the process of crossing over, independent assortment, and those types of things. We will create 4 haploid cells, each with 23 chromosomes if we're talking about humans, and we go through each stage of mitosis twice. So right now we're looking at the 2 cells. Each of them are going to go through the process of, or cell phase of interphase, which is the G1, which stands for first growth, the S phase for synthesis, where it's going to duplicate its DNA, as well as second growth, or G2, which is going to go through a second stage of growth, so that when the cell divides, it's not going to be super small. So here we're looking at interphase. All right, we got the centrioles, they're staying towards the middle of the cell. As we continue on with interphase, we are going to get to the first step of prophase. During this step, mitosis, those chromosomes are going to start to condense. You will see the centrioles start to go to either side, or start going to the poles of the cell. With meiosis, the homologous chromosomes, so the X's, are going to line up on top of each other. This is the part where crossing over can take place, because you do have sister chromosomes that are the exact same. They have the same type of chemistry, so there might be some switches that take place, and once again, something that adds diversity during the process of meiosis. Okay, so that's prophase. Once again, the condensing of chromosomes. Centrioles are going to start to go to the poles. Our next stage.
Call on line 1. Mrs. Holt, line 1.
Our next stage of the process of mitosis and meiosis is the prometaphase. Now, we didn't really talk about prometaphase, but basically it's a segue between prophase and metaphase, where they start to line up at the poles. We start to see the development of the spindle fibers that are going to start to move the chromosomes. Once we get into the full-fledged mitosis and meiosis, and we get to metaphase, then we will see the chromosomes actually go to the center. Now, some key differences here. Notice on mitosis that the chromosomes, the X's, are going to be lined up, and those chromatids, so a half a X, are going to be pulled apart. During the process of meiosis, the sister chromosomes are going to line up, but the full X's are going to be pulled apart, and that's going to have the number of chromosomes. So as we watch this now, okay, once again, we'll see these kind of being moved apart. Once again, this is metaphase. During anaphase, these chromosomes are going to split apart. Once again, here we have the half X's being pulled apart. Here we have the full X's being pulled apart. So that is anaphase. Once again, the key here is that these chromosomes kind of look like the letter A. That's the word hitch I use. You can use it if you want to. After anaphase, we then start to see the development of nuclei. Now, once again, if we look at the number of chromosomes, for both cells we started off with 4. All right, now if we count them, we still have 1, 2, 3, 4 1, 2, 3, 4. That's the mitosis one. Okay. Now if we go to meiosis, we only have 1, 2, and 1, 2. So during the process of meiosis 1, which is one sequence of event, the number of chromosomes are going to be halved. Because we only go through one sequence here, and it's the half X is being pulled, here we're going to maintain that chromosome number. All right, so let's continue on with our digital path here. Finally, during cytokinesis, once again, cyto means cell, kinesis means to split. So here we have the now two separate cells. With the mitosis, once again, we have two diploid, 4 and 4, and we started with 4. With meiosis, we have two cells with 2 chromosomes, therefore we have the number of chromosomes. So they're now haploid. So this is where mitosis now ends. You created 2 exact copies of the cells. The only thing here is they are now in their half X form. Now, as these cells start to go through their cell cycle, they will duplicate and then get the full-fledged X's again. So I kind of use this phrase often. It's almost like they double the amount of chromosomes they have before they have, so that maintains. Where in meiosis, it just cuts those sister chromosomes in half, and we now have 1, 2 chromosomes, so we already halved it. So mitosis is pretty much done. So as we look at this visual now, mitosis is going to pretty much stay put. During meiosis 2, it's going to go through the same sequence. So we have interphase 2. There we're going to have, once again, those chromosomes are going to start to condense once again. No crossing over during this portion. During prophase, once again, no crossing over because there's no sister chromosomes lined up. Metaphase, we're going to get these things. So there's prometaphase again. Metaphase, we're going to get these now X's on the equator. So these half X's are going to be pulled apart. During anaphase, the half X's pulled apart. During telophase, they start to develop 2 separate nuclei, and then the final part is cytokinesis. We now have 4 haploid cells from the original. So those are some of the differences, once again, between mitosis and meiosis. Mitosis, one sequence of those, prophase, metaphase, anaphase, telophase, where meiosis, you go through those twice. Here we have 2 diploid cells, exact copies. Here we have 4 haploid cells with some variation if crossing over does take place. All right, so hopefully this visual helped you out. Let's continue on with our next subject. Now that we have talked about how our cells have to duplicate their DNA before they divide, we talked about you have to double before you have. We're going to go through that process of how our DNA actually goes through DNA duplication. It's sometimes called DNA replication, basically means the same thing. Now with this process, there are 3 main enzymes that are involved. So what will happen is you will have a sequence of DNA. All right, and generally speaking, when we duplicate the DNA, it will go through the entire strand. The first thing that will come into play is an enzyme called helicase. And this is what kind of acts like an unzipper. What it will do is it will unzip the hydrogen bonds that are holding together the different base pairs. So G with A, G with C, T with A with T, so on and so forth. And it will unzip them. Now each of these is going to be the original strand of DNA. So helicase is going to do the unzipping. Then what will happen is you will have another enzyme called polymerase. And its job is now to use the original sequences, so the dark ones here, as a type of template in which to create the new DNA. So what polymerase will do is it will now match up, it will bring in these base pairs, so it's going to match up the G with the C, T with the A, etc., all the way down the line. So polymerase will attach those free nucleotides to that template strand. Now when you do this, you're going to create an overall DNA molecule that is half old and half new, but they're still the exact same. Nothing really changes with that. Very rarely are there mistakes that happen with this. Usually the process is pretty good, about 99%, 99.8% efficient, so very good at what it does. After you have helicase do the unzipping, polymerase do the attaching, you will then have ligase. And ligase is kind of the double checker. And what it will do is it will make sure that none of those mutations take place, like I just mentioned. So it will not only double check to make sure that all of these base pairs are lining up, but they'll also make sure that these things are completely sealed. So they'll double check to make sure the hydrogen bonds are together. They'll make sure that the phosphates and the sugar backbone are in place, and make sure, once again, that everything is in check. Once again, the net results are the exact same sequence, just now one is 1/2 old and the other is 1/2 new. That's kind of the difference. Once again, the whole process of DNA duplication or replication takes place during the S phase or synthesis phase of the cell cycle.
DNA replication begins when helicase unwinds a segment of the DNA and breaks the hydrogen bonds between the two complementary strands of DNA. DNA polymerase can only add new nucleotides to a free 3' end of a growing chain. Synthesis of one strand of the DNA, called the leading strand, proceeds continuously in the 5' to 3' direction. Synthesis of the complementary strand, called the lagging strand, is more complex. DNA polymerase can add new deoxyribonucleotides only to a free 3' OH. To provide a free 3' OH starting point on the lagging strand, RNA primase attaches to the DNA and synthesizes a short RNA primer. DNA polymerase III then adds deoxyribonucleotides to the 3' end of the RNA primer. DNA polymerase I replaces DNA polymerase III, removes the RNA, and replaces it with DNA. Finally, the enzyme DNA ligase forms a phosphodiester bond between the 3' OH of the growing strand and the 5' phosphate in front of it. During DNA replication, the leading strand is synthesized continuously, while the lagging strand is synthesized discontinuously.
We're now going to get into arguably the hardest part of our class here and talk about the processes of transcription and translation. All right, when we have this diagram, you have one similar to this in your book. So we just got done talking about the process of DNA duplication. Now, the DNA is what codes for everything that we look like, how we act, etc. We know that pretty well. Now what will happen is, if there is a particular protein that is needed by our cells, whether it's an enzyme or a structural protein, all of that is coded for in the DNA and by our within the nucleus. So what will happen is, the first step is the process of transcription. And transcription, in short, is going from DNA to messenger RNA. Now, when that takes place, it will be in the nucleus. And what's going to happen is, there will be a DNA strand that will unzip. Now, it's similar to DNA duplication, except you're not going to copy the entire sequence of DNA. Instead, it's just going to be a particular segment that actually does that. The enzyme that is in charge of doing all of the unzipping, the attaching, and then the gluing, so to speak, or the double checking, is what we call RNA polymerase. So during the process of transcription, it's when you go from DNA to messenger RNA, and it's all done by the enzyme, RNA polymerase. So that's what we call transcription. Let's see a quick video on that.
The synthesis of messenger RNA is called transcription. Transcription begins when RNA polymerase recognizes and binds to the promoter region on the double-stranded DNA molecule. A particular subunit of the messenger RNA, called the sigma factor, participates in recognition of the promoter region. Soon after transcription is initiated, the sigma factor dissociates from the RNA polymerase. RNA polymerase moves along the template strand of the DNA, synthesizing the complementary single-stranded messenger RNA molecule. Synthesis is in the 5' to 3' direction, with new nucleotides being added to the 3' end of the growing messenger RNA molecule. As the RNA polymerase advances along the DNA, it melts a new stretch of DNA and allows the previous stretch to close. When RNA polymerase reaches a specific sequence of nucleotides on the DNA called the transcription terminator, a hairpin loop structure forms in the messenger RNA, causing the RNA polymerase and the messenger RNA to dissociate from the DNA.
Now that we have our messenger RNA strand created in the nucleus, it will now leave the nucleus and go through the nuclear pores. Okay, those are like the little holes in the nucleus itself. That messenger RNA strand will travel to the protein-making factories, which we well know as the ribosomes. And this now starts the process of translation. And what it's going to do, and the reason why it's called translation, is that this messenger RNA strand now codes for particular types of proteins and amino acids that the desired protein is needed for. So what will happen is the messenger RNA strand once again will leave the nuclear pores and dock with a ribosome. It will start with the small subunit of the ribosome. The messenger RNA strand will kind of attach to it, and then this big or large ribosomal subunit will come into play and kind of attach itself. Then what will happen is the ribosome will read the sequence of base pairs in groups of 3. We call those codons. So let's just pretend that this codon is AUG. Then what will happen is ribosome will say, all right, I need the amino acid AUG, which codes for methionine. So this molecule, this is transfer RNA, will then bring in whatever desired amino acid is needed. So in this case, if we said it was AUG or methionine, it's going to bring in methionine. Now, the only other kind of weird thing with this is if this messenger RNA strand says AUG, that's a codon. Transfer RNA has what's called an anticodon, which is going to be the opposite. So if this was AUG, that means that this is going to be UAC, called the anticodon. Okay, you might be quizzed on that a little bit. So anyway, methionine will be attached, and any other amino acid will be attached to a portion of this transfer RNA. Now what will happen is, okay, once this codon is read, it will then slide, the messenger RNA will slide down, and it's going to read the next group of 3. Then transfer RNA is going to bring in that desired amino acid. It's going to bring it in. Every time there is a new amino acid that is brought in, it links up to the desired protein that is being created. This is oftentimes called a polypeptide chain. Peptide bonds are what hold these amino acids together. So collectively, when this protein is first starting off, you're going to get a few of these amino acids together, held together by a peptide bond. Once you get a lot of these amino acids together, it creates a polypeptide chain, and then we know that proteins have different types of structures from primary to secondary, tertiary, and finally quaternary. So that's kind of the overall process now of translation. Once again, it takes that code of messenger RNA, it's being read by the ribosomes, transfer RNA is going to bring the desired amino acid to the ribosome. The ribosomal RNA or rRNA is going to help attach these peptides together to create the full-fledged protein of whatever's being desired. Now, last little bit, one of the things that often happens here is if there is a mix-up of the DNA or if there is some type of mutation, the reason why that is so significant is that the simple changes in letters that can happen are very significant. So let's say that we had an AUG, okay? That's this one. I'm not sure if you guys can read that. No, you can't. Anyway, this one is AUG. It codes for methionine. Now, all of a sudden, let's say that there was a mix-up and instead it said AUC. All of a sudden, instead of having the right one, you might all of a sudden have something like asparagine right here, and you guys can't read that, sorry about that, but it's a completely different amino acid. So what that does is even a simple little change of the DNA sequence can completely alter the desired amino acids that are needed. Now, what you're looking at here, and this is on page 222 of your textbook, is you're looking at the different amino acids codons. So each group of 3 letters signifies for a different codon, and a different amino acid. There is one type of start codon. We call that methionine. That is a good one to know. There are also 3 stop codons that are found in cells. And once again, the 3 steps, if we go back, sorry, I'm jumping around here. If we go back to the process of translation, there are 3 main steps to this. We have initiation. That's where the ribosome will attach itself to the messenger RNA or vice versa. You then have elongation, and that's the process where the messenger RNA is actually going to be read. And then you finally have termination. And termination is when one of those 3 stop codons will be coded for, and that's going to stop the creation of those proteins. All right? So once again, the process of translation is now taking the messenger RNA sequence, bringing in the desired amino acids, and creating the proteins that are needed by the cell.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site, and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA, paired with the previous codon, is passed to the P site and then to the E site as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.